Okay, another quick chat here. All right. All right, let's go back to our. Okay, so that was one. Hopefully that's going to help you. Oh, damn it. A little bit. Treadmilling. So here's the idea of treadmilling, which is if you have a really high concentration of GTP tubulin, it's going to polymerize on both sides. If you have a really low concentration, it's going to depolymerize on both sides. But if you have somewhere in the middle where it's higher than the concentration needed to polymerize the plus end and lower than that needed to polymerize the minus end, you get depolymerization here and polymerization here. So while you're adding to one side, you're subtracting from the other. And blue here doesn't mean GTP, it means that's the ones that were added at that time point. And you can see they were added at that specific time point and they move backwards, 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 and eventually they'll come off. So the length of the microtubule isn't changing, but it's dynamic. You know, it's monomers are being added and subtracted, but the overall length is not changing. Now, skeletal muscle. So this is the other one that I recently uploaded. Again, I made this years ago, probably 10, 15 years ago, for your textbook. And probably if you have that, you can still find it there, but okay. So here's the idea. This I was talking about in class, the H zone, the A band, the Z line in the middle here, the I band from here to here, and the M, is there an M band? I think there's an M band up through the middle, but the point being the H zone is where you have only myosin. A band is completely of the myosin, which sometimes overlaps, sometimes not so much. And the I band is where there's no overlap of actin and myosin. So when these things contract, the little myosins grab and go like oars on a boat. Your H zone shrinks down. A band, of course, doesn't change because the length of the thick filaments doesn't change. The I band shrinks again because I band is where there's no overlap. And look at that. And where there is no overlap, decreases over time because the overlap increases. You can see there's a point there where you get more myosin heads working away, and so you get a stronger contraction at that point. Okay, questions about that? I don't think this works. Nope. Any questions about those animations? If not, I'm going to go ahead and do the exam.
For treadmilling, did you say it was the plus end that polymerizes or the minus end?
Right, the plus end. So that's kind of what they learned through this experiment, that, excuse me, the plus end is going to preferentially.
Yeah, wait, let me call you back because I just joined the Zoom for my class and I'm gonna call you when I'm done with it. Give me like, I don't think he's gonna be here for that long, so give me like 30 minutes. I'm gonna call you right back. All right.
Okay, Ashley? Okay, what was I saying? I'm sorry, I got distracted because Ashley, your microphone was on.
Sorry.
Oh, I love Zoom. Oh my God, Daniela's got a kitty. What's the kitty's name?
Her name is Lovely, but she swears she's like Spider-Man and she just hopped on my back. I'm just not.
Lovely, she is lovely. Okay, back to work. So what they learned through this experiment was that the plus end preferentially will polymerize because it'll polymerize at a lower concentration of GTP tubulin than the minus end. And so if you find that concentration between the two critical concentrations, it's going to just do this, okay? Let's give it up for Lovely. Yay, Lovely. All right, questions on dynamic instability, perhaps? Okay, again, that's about how GTP is important for stability. When it goes away, it depolymerizes. Oh, here's the other thing. Don't forget to do the game, right? Here's our game. Make it smaller so I can actually see it. Cross-linking proteins for microtubules, that would be our tau protein. Protein activated ICs and branching, that would be WASP. Adding subunits, we just treadmilling, a drug that affects microtubule formation. Taxol, drug that affects microfilament, phalloidin, enzyme that cuts inside the strands, gelsolin, causes nucleation, RIPS, an enzyme that depolymerizes, covalent. Check. I got them all right. So here's the thing too, once you do that, you can add the bonus, which is if I get them all right, I'm going to get 28 extra points. Okay. And then I'm going to go through here. The next one is a drag and drop, which I'm not going to bother doing. And if you get it wrong, the right answers come out so you can learn how to do it for the next time through. Here's another kind of word drag and drop. And another picture drag and drop. Oh, I hope I have this right. What is this? Let me make sure I have this right because I screwed it up at one point. That is not tropomodulin. That is, oh man, I don't even know. Good Lord. Oh, there it is. Tropomodulin is there, I believe. Cap Z is here. Nebulin and Z line is here. Thin filament, actin, thick filament, myosin, troponin, titin. Oh, this is titin. Let's do this. I don't even know if I'm right. Oh, why do I ever doubt myself? I'm so seldom wrong. Add bonus. Next. Okay, and then when you get here, take a screenshot of this and send it to me. Although, you know, at 194, I didn't get any bonus points. So that's to encourage you to do it again and again and again. And that's going, and you're going to do it again by hitting refresh. And that's to encourage you to do it over and over again until you get a better score. And when you do, you're going to learn this stuff better. It really helps to get this crap into your head. And by the way, the questions scramble every time and the answers scramble every time. So you can't memorize where the questions and answers are. Okay. Chat. What's the highest score? I've had people get 12 points on it before, so I don't know how, because there's no way I could do it. But some people are into it. And again, you repeat it a lot, it gets easier. The more you repeat it, the easier it gets. Okay, draw or describe the structure that attaches the cytoskeleton to the plasma membrane. So this is pretty easy. Let me just pull up my PowerPoint and I'll show that to you. Okay, so this, am I sharing this? I don't think so. So this is what I'm looking for, that question. Your band 3 and chirin is attaching spectrin to band 3. And then you've got band 4 and a microfilament over here, and there's glycophorin. So again, you know, with these questions, I'm just, you know, it's just memorization. If you memorize all the stuff, then you'll get those questions right. Okay. Fill in the blanks. A protein that binds to microfilaments to begin a new branch. That's ARP23. An enzyme that depolymerizes microfilament ends, that's ADF or cofilin. G protein responsible for stress fiber formations, that's RAC, R-A-C, RAC. A drug that stabilizes microtubules, blocking them from depolymerizing, that's Taxol. An enzyme involved in phase transitions of amoeboid movement, that's gelsolin. Phase transitions, meaning going from gel to sol, gelsolin. Calcium binding protein that controls tropomyosin position, that's troponin C. Transmembrane proteins that bind to extracellular matrix, to this, transmembrane proteins that bind extracellular matrix to the cytoskeleton. Oh, that's the integrins. Oh, wait, extracellular matrix. Yeah, integrins. Motor proteins involved in moving the cilia, that's dynein, your axonemal dynein. Okay, fill in the blanks of sarcomere structure. Ooh. So there should be a picture here. There is not. But if we go to the answers, you can see the picture. Okay, so there's a picture for one. Okay, and this, someone pointed out to me that it was RAC row. Molly, you're right, it's row, it's not RAC. Yeah, so the question was stress fibers. Stress fibers are activated by row. Well done, Carrie. Okay, so back to this. So this is the answer for number 3, tropomodulin, nebulin. I had those switched originally. Thin filament or actin. Cap Z is the protein here. This is the Z line. Titin, myomasin, thick filament, or myosin. Describe or diagram the process of amoeboid movement. So we talked about this yesterday in class. It wasn't yesterday, was it? It was 2 days ago. So it's just the process of microfilaments being depolymerized at one end, being forced through the gel actin or polymerized actin, and then forcing the plasma membrane out and then polymerizing here. That's amoeboid movement. And it's being, the push is because of myosin, which is causing some stress here that's forcing the endoplasm through out the other end. Okay. So again, those questions are just memorizing what I did in class and giving it back to me. All right, now more complicated. I think we did one this or one like it in class. GTP gamma S is a GTP analog that binds to the GTP binding sites. It will substitute for GTP, but isn't hydrolyzable by most enzymes, including tubulin. If this is added to treadmilling microtubules, what would likely be the result at at each end? Okay, so the basic idea is that if GTP is hydrolyzed to GDP, that will destabilize the structure and you get catastrophe. So even if you've got a high concentration of GTP and tubulin there, you're going to get dynamic instability, which means it'll grow and shrink and grow and shrink. But if you can't hydrolyze GTP to GDP, it's only going to grow, and it's going to grow at both ends. Or if the concentration is lower than the critical concentration for the minus end, it will the minus end will be stabilized and the plus end will grow. So, you know, it depends on what the concentration is, but I would accept any answer for that. And again, here's the idea. Like the idea behind the question is, what does GTP do in microtubules, right? So, no, I didn't say it there. So if you know what GTP is for, what happens if it can't be hydrolyzed? Okay, that's how you get to that question, that answer. Okay, researchers who studied skeletal muscle contraction developed a model system called the skinned muscle prep. In this prep, a single muscle fiber, i.e. one cell, is dissected away from its bundle. the fiber is put into this is true, actually. They used to do these experiments. The fiber is put into a physiological solution and treated with detergents to remove its cell membrane. This exposes the sarcomere to the external medium. Given this prep, what two things would have to be added to the solution to cause the cell to contract? Explain how each is used by, it should say by the tissue, not but the tissue. Big dope. If I could spell, I'd be dangerous. Okay. Oh, stop already. Let's not save it. Okay. So the idea is, okay, so we talked about excitation contraction coupling, right? But excitation contraction coupling all has to do with the membrane, you know, the action potential coming down, the T tubules going down the T tubules, the carbon, the calcium channel interacting with the ryanodine receptor. Ryanodine receptors in the sarcoplasmic reticulum, but that's a membrane too, so that's gone too. There's no membranes at all here. All we have is the sarcomere. What's it going to take to make that contract? Well, you need two things. One, you're still going to need calcium to move the tropomyosin off the myosin binding sites. Once you do, myosin can interact with it, but it needs actin to contract. So all you really need at this point is calcium to activate the thin filaments, and ATP to allow the thick filaments to start to contract it. Okay, so again, how do you get to that answer? Think about excitation contraction coupling and take all the membranes away, and what's left? There's nothing left except the contractile apparatus. What does that need to contract? It needs the thin and thick filaments to be able to interact, which is calcium, troponin, tropomyosin, that whole schmear. And then it also needs ATP in order for the energy for the contraction to happen. Okay. You are a cell biologist. Hey, it could happen, right? It could happen. You are a cell biologist studying the crawling behavior of fibroblasts. Now, when you read that, immediately you're going to say cell crawling. What do I know about cell crawling? Okay. One of these cell lines that you are studying has one mutated gene. Okay, so there's one protein that doesn't exist anymore. It's non-functional, it's not there. These cells look structurally normal, however, they differ in their response to PDGF. So PDGF is your chemoattractant that's going to cause these things to start to move, but they're not acting normally here. In response to this hormone, cells are able to extend filopodia, but are unable to make lamellipodia or to move. What protein may be mutated? So what do we know about lamellipodia? Right? That's what the question is asking. What does it take to make a lamellipodia happen? And what individual proteins are involved? Probably the most important one is RAC, right? We saw in that one experiment that RAC causes lamellipodia to form. So if the cells were lacking RAC, then they wouldn't be able to do that. So normally PDGF would activate the receptor, which would activate RAC, which would then do that PI3 kinase thing, and then you would get the lamellipodia to form. Now, if you go downstream from that mechanism, you've got PI kinase, right? So maybe that's gone, because if RAC activates, but there's no PI kinase, well, then the whole thing stops right there. Another thing that happens is CD42 is important for the branching of microfilaments to make that lamellipodium. If CDC42 isn't there, then it can't happen. So, basically you're talking about most likely RAC, but it could also be PI kinase or CDC42. Although frankly, if you didn't have PI kinase, there'd be a lot of things wrong with that cell. It's a really important enzyme, but you wouldn't know that. So it doesn't really, I wouldn't take points off for it. Okay, what if it couldn't extend filopodia? Well, then CDC42 is important. What if it extended a lamellipodium, but it wasn't able to crawl anywhere? It just sat there with the retrograde flow of actin going. Well, then it's not attaching anymore, right? So there must be something wrong with the attachment. What happens with attachment? That's integrins. So that could be what's wrong. So again, the way you approach this question is, reading it, cell crawling. Okay, I know what's involved in cell crawling. You've got 4 different steps, and I know what proteins are involved in each step. And it is able to do this and this, but not that. Well, which of those proteins were important for that? That's probably what's screwed up. And again, it's hypothesis generation, because it could be a lot of different things. And for questions like this, a lot of times there's multiple answers that will work. They just have to make sense with the experimental data that you're given there. Okay, questions, comments, discussion? No discussion. I feel like the older I get, the more I talk. When I was a young professor, my classes were always short because I never knew what to say. Now I can't shut up. Okay. Oh, here's something I love to do, frankly. The day before the test, I'll scan some journals to find out what's new. What did we just learn about what has just been learned about the proteins and processes we were just talking about in class last week. And then I'll put it out, and basically all I'm asking you to do when I do this is to read that paragraph and explain it. Okay, so I'm not asking you to add anything, but just use the knowledge you have already to explain this paragraph. So what the heck is this about? Homeostasis and wound healing rely on stem cells whose activity is and directed crawling are often governed by chemotactic signaling. Okay, we're talking about cell crawling again. What do I know about cell crawling? Okay. In dissecting how this pathway integrates with cytoskeletal dynamics, we discovered a kinase that is inhibited by chemotactic signaling, directly phosphorylates spectroplakin. Okay, so a kinase that is inhibited by signaling phosphorylates this protein spectroplakin, a 500 kilodalton microtubule actin cross-linking protein abundant in stem cells. Okay, so what is that saying? Kinase is a phosphorylating enzyme. So when the cell is not moving, then spectroplakin will be phosphorylated. When you inhibit that kinase, it'll be unphosphorylated, and then it can crawl. That's just the logic of the sentence. You don't need to know anything except how to read that sentence. We map spectroplakin's phosphorylation sites to the microtubule binding domain, and show that phosphorylation uncouples spectroplakin from microtubules. Oh, we're tying microtubules into it. So it sounds to me like, again, I haven't read this in over a year, so it's fresh for me. We map spectroplakin's phosphorylation sites to microtubule binding domain. So it sounds like when the cell's not crawling, spectroplakin is cross-linking microtubules with microfilaments. Mutant spectroplakins that are either not phosphorylatable or constantly phosphorylated inhibit movement polarized towards the chemotactic source. Okay, so if spectroplakin phosphorylation can't be modulated, then the cell can't move. Revealing that phosphorylation must be dynamic. So it must be phosphorylating, unphosphorylating, phosphorylating, unphosphorylating. Because in both states, both states are important for the movement. This circuitry is physiologically relevant. Oh, it's physiologically relevant. That's good to know. And depends upon polarized inhibition of this kinase, polarized at the front of the crawling cells during wound repair. Okay, so it depends upon polarized inhibition of the kinase at the front. So at the front, you're going to be inhibiting the kinase. At the back, you're not. Okay, so it's polarized, and that is going to direct the cell in the direction of where it is inhibited. What is the normal role of spectroplakin in cytoskeleton? So it sounds to me like it is cross-linking microfilaments with microtubules, right? Microtubules, so it binds to microtubules. So it's going to kinase is inhibited, directly phosphorylase a microtubule cross-linking protein. So it's cross-linking microtubules. Oh, so it's not binding to microfilaments. It's cross-linking microtubules. Okay, so that's what it is. And cross links my MTs. Now that answer, I didn't make that up. I just read that. Okay. Signaling directly phosphorylates spectroplatin, a microtubule actin cross-linking protein with. So again, here's the thing with these kinds of questions. I'm not asking you to do a whole lot more than read the paragraph. What is the normal role of spectroplatin in cytoskeleton? Spectroplatin is a microtubule actin cross-linking protein. Okay. Describe the role of spectroplatin in polarized movement of stem cells during wound repair. What is the role of spectroplatin? So what is the role? I guess I would say the role is that it becomes it allows, what is the role of spectroplatin? Polarized directed movement of stem cells during wound repair. Roll my sleeves up. Spectroplatin cross links the MTs and MFs when active at the leading edge of a crawling stem cell, comma, crawling stem cell. It is inactive, which allows the MTs and MFs to move. Booyah. That's about it. That's about all you'd know, right? You don't want to go beyond what the paragraph actually says. I've done a little bit of interpretation here, but mostly I'm just saying what the paragraph says. Spectroplatin cross-links them when active. The leading edge at the trailing edge, they remain attached to each other. All right, that's what I think. Let's look at the answer, because I've been wrong before. In polarized chemotactic movement, one side of the cell has signal transduction activated by the chemotactic source, while the other does not. When spectroplatin is phosphorylated, it releases the microfilament. The kinase that phosphorylates spectroplatin is inhibited by the chemotactic signal. The phosphorylation dephosphorylation of spectroplatin is important for cell motility. Therefore, the attachment detachment of microtubules and microfilaments by spectroplatin must be important to cell movement. That makes sense? I'm doing a lot of talking, nobody else is. Okay, in this little reaction thing down here, reactions. Give me one of these if it makes sense. And what else do we got? We don't have a thumbs down. There's gotta be a thumbs down. Do one of these if it didn't make sense. React now. Everybody react.
React.
That was Dylan. Thank you for reacting, Dylan. I think. Okay. Serby, are you sleeping? Are you still there? Serby's gone. Willie. Willie Corley. No. Sukata is not sure. Sarah Krausnick is in.
I'm sorry, I'm trying to pay attention. When I woke up, my bank account was empty, so I'm trying to do that.
Damn. That's just sad.
And complicated.
Yeah, yeah. Well, go talk to Dr. Buckley. I'm sure he'd lend you 20 bucks if you needed it. He's that kind of guy.
Thank you. I appreciate that.
Okay. So that's about all I plan to talk about today. We still have time to kill if you have some questions. So let's open this up to the floor. Anybody has a question? Go ahead and shout it out. You've got questions, I've got answers.
So the exam is on Tuesday.
Tuesday at 11, same room.
Okay, and the game extra credit just has to be done before that.
Right, exactly. Has to be done before it. My whole point of the game is it's going to help you do better on the exam. So it's not really worth it to do it afterward.
Yeah, absolutely.
And so the game thing is timed,
Yes.
and like after that it doesn't count, like if you take too long to answer.
No, the time is only for bonus points. So if you get all the
Oh,
right,
okay.
yeah, if you get all the answers right, you'll get 80 points per, is it 80? I forget. You get a certain number of points per level. And I think the points double if you get them all right. And then the bonus points double if you get them all right. So, the better you do, the faster you do, the more bonus points you get. But, you can take your time, especially the first times through. Don't worry about the timer,
Okay.
especially if English isn't your first language, I'm guessing.
Yeah, no.
It's harder, I know. What language do you speak in Azerbaijan?
Azerbaijani, our first language.
That makes
Yeah. It's kind of Turkic, similar, but not the same.
Okay. Well, I don't speak Turkish.
I know this is going to be a dumb question, but I don't know why, but exam is on Tuesday or Thursday?
Tuesday.
Tuesday. Okay.
I don't know. Does it say that on the syllabus? If so, somebody got that.
Syllabus
syllabus
said
it
the
says Thursday.
24th.
You lie. I'm just kidding.
Right?
Syllabus, I often just roll it over from
Yeah.
one year to the other. So sometimes I make mistakes.
So it's not the 24th. It is the
No, it's the
22nd.
22nd.
Okay.
Yeah, I'll fix that today and get it back up. Everyone is on board with that. Sorry.
No, I just was confused. Okay, Tuesday.
Okay. This exam, I don't think is the most difficult one. For me, this material is not as interesting as other stuff, but I don't think it's that hard. When we get into the third exam where we're talking signal transduction, that's a little more complicated and a little more interesting. Yeah, the next exam is kind of like the first one too. So any questions? Jeffrey Cusey, I know you've got 100 questions. Just pick one. Philip. Oops.
I think I'm good.
You're good. We'll see Tuesday, won't we, mister?
Yep.
All right, well, we don't have to sit here if we're all good. Tell you what, if you've heard all you need to know, go ahead and leave. If you want to stick around and talk some more, I'll stay here and I'll turn the lights out. I'll be the last person. Good luck.
If we're
people.
not good, unrelated to cell biology.
That's fine. I do
just-
relationship advice.
I'm just kidding. Thank you.
Sure.
Have a good day.
You too. Take it easy, Dylan. By the way, I could be bought for beer if it's good beer. Go home and pray. Praying's not going to help you, Philip. Have a good day, Beamlek.
Where did I hear that recently? That I could be bought for beer, but it has to be good beer.
I don't know. I thought I made it up.
No, I swear to God that had to have been in a show or something because I literally just heard that recently. And it kind of freaked me out that you said that because I was like, wait, what? Also, I was curious to see who would end up staying.
No, I'm Googling it right now to see if it is from a show or something, because I
Okay.
felt like it was some like familiar too.
Are you Googling good beer? Because you could look up Sip of Sunshine. That is a good beer. I'll be drinking some of them tonight, I hope. This is my Friday, I don't go in on Fridays. So Thursday night is my Friday night.
See, I only have school on Tuesdays and Thursdays, but Monday, Wednesday, Fridays, I work at the toxicology department. So
Uh, so you don't get off anyway. All right, well, I'm just gonna close this all up unless you've got some cell biology questions or relationship advice. I do that too.
Oh,
wow, just the two? That's all? You're not even gonna wait for us to spoil that you stole that from a show?
Okay, I gotta wait for that.
I couldn't find anything. I didn't find anything.
I'll find it.
Maybe
Come
the
on.
show stole it from me.
You-
I can show a really cute thing here. So this is the blanket. And my cat decided to come and sit with me today. I don't know if you guys can see him.
Oh my God, what's his name? Her name? Its
That's
name.
Perry.
Hey, buddy.
He
Hey.
likes to cuddle in when I have my Zoom classes. Every single time he comes in with the blanket and listens. I feel like he's smarter than me at this point.
We had a
what's
big-
it called?
We had a big,
Go on.
our cat's about 20 years old and he's thin as a bone. And he's always trying to sneak out of the house. And so we try to keep him in. And he's been missing for a day and a half. We're like, uh-oh, we don't know if he's in the house dead somewhere or outside dead somewhere, but we haven't seen him in a while. Showed up shaky this morning on the back porch. Let me in. So he's still
Oh
alive.
my God.
Yeah, he's
My
still
Lord.
alive.
20 years.
I felt bad because you guys were showing off your animals and I didn't get to show off Z.
Oh.
How do I flip the camera on this? Oh, there we go. My chinchilla.
Oh my gosh, that is so cute. Oh, chinchillas.
So here's something you might not know. Plattsburgh State used to have a chinchilla colony. They were doing hearing research. So they were doing research on chinchilla ears. Yeah.
I actually wanted to, I was so curious to like what frequencies he can hear because I always play music in the car and he doesn't seem to mind it, but I don't want to hurt his actual ears. So I kind of get a little like iffy about it, but I did not know that they ended up doing research on that.
Yeah. Oh, absolutely. They got grants from the US Army because they were trying to find drugs that would protect ears from damage. Because, Army, there's a lot of banging and booming going on. But at the
Yeah.
same time, you don't want, if you're in the Army, you don't necessarily want to have your hearing covered because you want to be aware and alert, but you don't want your ears destroyed. So they did a lot of work on that.
Did they end up figuring out something or no?
I don't think so, but they got some money.
Well, at least they got some money.
Yeah.
All right, well, I'll let you leave. I couldn't find the quote. I'll probably come to class next time with it.
Okay.
I swear to God, I heard it somewhere too, unless one of my friends said it to me recently, which in that case, that would just be weird. Yeah. All right. Well,
Could be.
have a good one.
Okay.
Bye-bye.
See you later.
Bye.